In this first part, we're going to look at some very basic exam tips, okay? So it's very important, and I'm going to go through this document, which you have in your classroom so you can slowly walk through, but I'm just going to talk through some very key points from this. So it's very important that you know your exam and the exam structure, okay? Because the papers are designed in a very specific way. So this way is like an increased level of difficulty because of course you start easy and then you get into the into this like focus mode, okay? So following the order of the questions is not a bad idea because you will see that you get to like, you know, the further you go to the paper, the higher the standard, okay? Which also means when you do exam practice, think about it. Maybe don't always start from question one of a paper, but when you do just exam practice on your own for revision, maybe check out the final questions of the paper so that you get these bit more difficult ones, okay? Because imagine if you just start a paper and then you leave it there, you only get the easy ones. So pay attention to that. But the most important part I want to discuss today is this objective. So there are specific objectives assessed in the paper. Each question is designed to test one of those. And when you look at the mark schemes, sometimes, or at least in most mark schemes, you do have the objective and the specification reference. So if you don't know which topic is, this is the number that you need to look up in your specification, okay? Also the type of objective. Let's look at this. The first one, which will be like 40% of your paper, is relatively the simpler ones, like you demonstrate knowledge and understanding of scientific ideas and techniques and procedures, okay? So this one, it's like comes straight of the knowledge, from your knowledge. I'll show you example of questions. Compare the structure of an artery with the structure of a vein. Notice that it's something that you study, okay? And it will be, notice, like some bullet points specific from the knowledge. Now, you needed to look at the type of question. So I do have a list of command words at the end of the document, which is really useful. But you have all of this like compare, describe, discuss, okay? Something straight of your knowledge. When you're studying things like structures, cells, any organ, it's very useful that you study it and you practice it. You can even like do this active recall way that you just take a diagram and describe it. But I think it's very nice to do it in a comparison way. Okay, so it's very useful because if you just, for example, you needed to do the structure of the heart and you just like repeat the structure. But then when you're asked to compare the structure of the right and the left-hand side of the heart, it's actually a lot more difficult, right? So whenever you can make your active recall, your revision, a little bit more challenging, do it because it helps you then applying it to exam papers. Okay, so instead of revising just the topic like this, make it more difficult for you. What I mean is, like doing in a comparison way and so on. Yeah. So, and that's the tip for just revising. And then you have this document so you can try these questions. Okay, now, and here I'm going to do something because those questions are the ones that I want you to actually try out. So, now, the important part of this is the second objective, which is apply knowledge and understanding, scientific ideas, inquiries, techniques, and procedures. So those are the more difficult questions. What it means is apply knowledge. It means that you could be given examples that are like that you're really not familiar with. You know, those examples that you're like, wait, what is, like, I've never heard of this before. Or like they give you a random name of a bacteria and you're like, whoa, I've never heard that before. What is it about? Okay, remember, they're testing this objective, which is the apply knowledge and understanding. So it always down to like a basic concept that you've studied. That's why it's so important that you have the overview of all of the topics, because you can always link back to that specific topic. And then the knowledge of the topic, you can apply it with the information that you're given. Also, if it's a question that it's very difficult and you're very unfamiliar with, you're given a lot of background information. Okay, so you would notice, and then once you see the structure of the question, like, oh, wait, it could be this. So let me not get thrown off completely, from this question, because it's this objective, which means that you should, take a bit more time to interpret it and to link it to a topic that you know. Okay. For example, now we're going to have a look at a couple of these questions, just so that you see the more difficult side, like this one. So for each question, also how it works, if you're watching the recording, you have to pause the video and attempt this question. And if you're live with me, you will get a couple of minutes, or depends on the marks, 1 minute for 1 mark, and you actually can try it. So now it's time to do that. The point of this type of questions is that it's not straightforward. You will have to apply some knowledge of your understanding, as the objective states. Remember, it's the hardest objective that it's tested. Unfamiliar topics, unfamiliar links between the topics. For example, this one. A person with AIDS may take longer than an healthy person to recover from a salmonella infection. What does AIDS have to do with salmonella? You haven't studied this way. So you really have to retrieve the knowledge from your knowledge. So I would suggest that you always look for the keywords that are relevant in the question, because these will link to the topic. And in this case would be, well, what did you study? You studied the immune system, you studied also AIDS. So how could it be linked to, recovery of an infection? Salmonella doesn't matter. It could be any infection. Okay, it's important to notice that too. Sometimes they give you information that are not relevant. It doesn't matter if it's salmonella or if it's any other infection. So the link here that you should mention, and I'll show you the mark scheme, would be, well, the immune system is damaged. It's weakened or doesn't function properly because, what is it? AIDS is an autoimmune disease, which affects the immune system. Therefore, the white blood cells cannot kill the bacteria. Do you see how it's like a consequential thing? And those are the nicest questions in biology, really consequential. Okay. And then, as you can see, it's not straightforward. So the other type of questions and the other type of objective would be analyze information and ideas to interpret, evaluate, make judgment, draw conclusion, experimental procedures. And this is what we'll practice a bit more today. Here there is another example of question, but we're not going to do it now, okay? Because we're going to cover first some investigations that are important, okay? So as I mentioned before, keywords are very important in your papers because it gives you an idea of what you have to do and also the type of question, okay? Remember, when you have suggest is the tricky ones, okay? Explain, it depends. It could be explanation of a very direct concept, or it could be one of these trickier exam questions, okay? So this is all very important. The type of question you come across, I would invite you to have a look at this. Actually, let's try one more question, because I can see that we have these nice objectives that we can test, because once again, these are the hardest ones. So we're gonna do one more question, and then we're gonna get started into. Okay. I think this is very nice. And don't look at the answer. I think it would be, oops, wrong color, but doesn't matter. Let's go with yellow. Let's give it a go with this one. Okay, notice it's an explain question, but it's still not straightforward. Very likely that if it's this objective that I said it's difficult, you're going to have an unfamiliar concept, like an unfamiliar example or something. So let's give it a go to this one. Pause the recording if you're watching, or 4 minutes for all of you. So this question, it will test some of these objectives, because like leaking heart valve, well, you haven't studied a leaking heart valve, but what did you study? what is a heartball, and what is the function. So that's what you will need to link to the cons. Then here is the mark scheme. So make sure you check if you got some of these points. Notice also that level type of questions, so 4 marks and above, they want relevant points, of course, with reason and causes, logical and clear format, okay? Don't worry too much in the super prose and perfect English. Get the points, okay? Here, in I would say things like, okay, the backflow can occur, less blood around the body, less oxygen supplied to the cells, therefore less aerobic respiration, example. Okay? Or you can add many other points. Okay, notice for 4 marks. Just think about the key ones. But notice how it's such a consequential question. Okay? Remember to check also your marks. It's extremely important because like this, you notice how to improve, which mistakes do you have? Where do you lose marks? Okay? Is it maybe one more point missing? Or maybe you didn't explain something. Okay? So the next part will be about the practicals. Okay, it's minimum 15% of the marks, so it is a lot. Okay, now you could be asked several things like constructing an hypothesis, plan a method, safety, understanding all the variables, reading scale, errors, data analysis, improvements. Okay, I think improvement is a very challenging one because you needed to be very familiar with the practical itself, and then you needed to think about how to improve it. But there are some general ways to get these marks, which we will see soon. Now, for revising practicals, my tips is to make it very visual, okay? So make sure you impersonate yourself in the lab or wherever you're doing or you've done the experiment. And like this, you would be able to describe it in more details, okay? So also, yeah, that's what I'm saying. Imagine yourself in the lab, walk through every step you take to perform it, okay? It has to be very specific and you would need to understand fully what is being investigated. Okay, I'm going to move further into, okay, never mind here. We are now going to look at some actual practicals. Okay, does anyone have any questions? But did you understand? I hope you understood the point of testing some of the more challenging objectives, which is that second objective. And now we're going to look at some investigations. Okay, so everyone, first of all, I see that not many of you are in the classroom. I will send the link right now in the chat to everyone. Please join right now. Because here you will have all of the points. Okay, let me stop here. Sorry, 1 second, I'm going to stop sharing, close the screen. We're going to revisit 3 key practicals and then get exam practice for each, okay? I've selected this because food test comes up in, it's very common in every paper, plus it's very linked with enzyme activity. Second, enzyme activity because enzyme is really one of the most important topic, and photosynthesis because also extremely important for the whole plant section of the specification, as well as a topic itself. Okay, remember that you have the topic list here, so whichever your specification is, make sure you look at that, because I have organized all of the topics for you, depending if you're triple combined, so that you can get a nice idea of your paper. It's extremely important that you look at what is assessed, how it's assessed, and then you have the topic list with the specifics of the practicals as well. Okay, so you have a nice summary to test and also to have it as a checklist before and after, your revision. So it's very important that you start this way. Okay, now we're going to start with the practicals, and what we're going to do is to start with, we're going to have a look at the content. Okay, so This topic, so food test, is extremely, sorry, is extremely important for biological molecules, okay? So notice how it's tested, like it asks already, like it mentions already the concept of enzymes, okay? So enzymes, extremely important for digestion, but also in all the other metabolic reactions. Okay. Anyway, after and in your own time, I would like to go through these reflections, okay? Because all of these could be like exam questions. So these could be exam questions that come up. So it's extremely important that you do this reflection, but for every practical that you come across and revise, okay? Because this will help you, being a bit more aware of some key concepts, okay? Now, the practical itself. When we look at tests, and food tests specifically, we need to prepare the sample, okay? So when you have to design an experiment, this is something to not forget, okay? You must prepare the food samples, and here, depending, of course, on the food you're given, you need to specify differently, but would be, for example, breaking it up, like with a pestle and mortar. If it's a liquid food, maybe you need to transfer it, okay? It's very important that you adapt this to the type of sample you're given, okay? But this is common for every practical. Think about the sample itself. Do you need to do anything with it or not? Now, to go very briefly with the test, just to revisit them. Test for glucose, it's a reducing sugar. Remember, Benedict's solution, this is the one that you need to heat up. Color, orange to red, very important. Second, starch, it's iodine. Okay, iodine is the food sample, you had iodine, eat, positive result would be blue-black. Third, protein food test. This is the purette solution. If it's positive, it would go blue to violet. If it's negative, it stays blue. Lipids. So lipids is the emulsion test. Okay, remember, we have ethanol, you add ethanol, and here, I mean, it's just a relative sample, but notice how stating the volume, it's also very nice when you describe an experiment. Anyway, you would, you could do it like that, allow time for the sample to dissolve in ethanol, strain the ethanol solution into another test tube, and then you add water to it. So you add ethanol, yeah, you strain it, and then you add water, because this step would form a cloudy emulsion if you have a positive test, because it's like the fat present in the sample that it stays a solid, it forms this emulsion, okay? Important to have a little summary. So when you have something like this that is like you really just have to remember from memory, make sure you have some nice notes out. That's why I've shown you this note with diagrams, and also this could be a table that you can copy down in your notes or make it as a revision card, because it will help you also when we apply to exam questions, okay? Now, some of you may be familiar with the CORMS evaluation, some of you won't. But I still think it's one of the best ways to do an investigation and to make sure you get all the points down. So always think about the change of what you're investigating, the organism, so whatever you are investigating, which is the sample. Repeats always must be mentioned. The measurement, how you will measure your dependent variable, what time scale you will use if that's relevant, and then the same, which is the control variables, okay? Some of you may just be familiar with dependent, independent control variables. This is another way to make sure you always mention the repeats and always think about your sample, okay? So for who is ever doing this CORMS evaluation, you can go back to the recording and check the CORMS for this. Otherwise, we'll go straight into a couple of questions, okay? We have the mark scheme over here, but just so that you see that, well, okay, sure, it's very simple, the topic, but then can you apply to questions? Let's try. Pause the recording if you're watching and give it a go. Okay. Otherwise, 1 minute for all of you to practice this. If you've done already, please raise your hand so I know. The solution. Okay, so the first one, as you notice, is glucose. So it's going to be Benedict's. Let me type it down over here. It's going to be Benedict's. Reagent is added to test tube. The tube changes to a brick red color. Part B, the correct answer is A. Okay. Background information here. Notice, once again, this is like a bit of an application of the knowledge. Second part. Okay. Okay, we are at this quest part 2 and part 3. Let's give you 3 minutes to complete it. So this is just a nice example of an investigation, yeah, where you don't have, like, you haven't done this experiment, but it's relative to one practical, right? One required practical that you've done. And as you can see, you have enough information for you to be able to interpret. And then you needed to do some like explanation. And for example, improve the investigation. I'll show you right away the mark scheme so that you can check your answer. Now, so conclusion, justification, reasoning. And you could say something around here. Explain, as long as you justify your answer, you may be given the mark. Not, for example, in this specific case, milk does not change the mass of the animal, or a milkshake reduces the mass of the animal, okay? So therefore, milk causes less tooth decay. If you got only 1 mark, it could be because you didn't think about this so what. Whenever you feel like you don't have enough marks, ask yourself, so what? This could help you putting, like, you know, giving your answer another layer, okay? 2 ways in which scientists could improve the investigation. Now, the improvements. We can always think about a few things. The instruments, make them more with a higher resolution, for example, or the sample that you're using, or the control variables that you should control. In this case, notice, control variable would be like the, actually here it's about shorter times repeatedly, in sense that instead of having like a longer time, you investigate it into shorter intervals. It's kind of a control variable because you are controlling the time, but you control it better. And then the sample that you're testing, okay? Always saying the same thing. Could be this, in this case, real teeth, clean the teeth, the same teeth, you know, something about that. If you have a human, you would have to say controlling some more variables of the humans. If you have a plant, it like could be the same plant, the same type, same age, all of these things, as long as the sample that you test, it's the same, unless that's the variable you're changing. So improvements are closely linked to that type of investigation, but we can always mention something about instruments and the sample and the control variables. Okay, I hope it's all clear. And now we're going to the next one. The second practical that I want to do is about enzyme, because it's very important, such an important topic, okay? And there are some keywords that if you just learn this, somehow you're gonna get some marks in this paper, okay? That's why I would like to focus a little bit more on the specification content, okay? So that you can refresh this as well. So enzymes, explain the mechanism of enzyme action, active site, and enzyme specificity. What comes to your mind when you think about this? The whole enzyme activity topic, okay? It's very, very important. And it's extremely important to mention the fact that the substrate and the enzyme active site, they're complementary. So this concept of complementarity, it recurs in many topics in biology, okay? So remember that the mechanism of enzyme action is biological catalyst. How does it work? Enzyme and substrate, they move in the solution, yeah? Then when an enzyme and the complementary substrate randomly collide, this complex enzyme-substrate complex forms and the reaction takes place, okay? Product is released from the active site. Now, one mark when you have to explain this, one easy mark that you can lose is to say that, well, if you think about the substrate, it's complementary to the active site. But once the product is formed, the product is different than the substrate, so it's released because it's not complementary anymore, okay? So as the product is formed, it's released because it doesn't fit anymore in the active site. Very important, the enzyme is unchanged. So this is the mechanism. Okay, you may have studied it as like some different actions, the induced fit model or the lock and key, but just be able to describe it, and this fact that it's complementary is always very important. Substrate, active site are the keywords. Second point, explain how enzymes can be denatured due to changes in the shape in active site. Once again, think about how you can say this in a few marks. Very important, okay? Because you need to use the correct wording. So denatured, it's extremely important because remember, enzymes are proteins. Proteins in the biological molecules topic, proteins, they are very sensitive to the environmental conditions. So when the conditions are outside from their optimum range, they denatured. What it means, change shape. That's the link that you have to make sure you mention in an exam question. Denature, change shape. If the active site change shape, the substrate is not complementary anymore. Remember, it's always a consequential thing in biology. So this is the second important point. Third important point, effects of temperature, concentration, and pH on enzyme activity. Temperature and pH, they are related to these to the fact that proteins and enzyme act at their optimum ranges. So above or below a certain temperature, so if it's a too low temperature, the collisions of the particle is too slow. So the rate of reaction may be too slow. If it's too high, the protein denatures. So once again, the active site denature. Same for pHs that are above and below and below the optimum activity. But if you think about what about substrate concentration? It's once again because of this collision theory. The higher the substrate concentration, the more collisions there are, until, of course, all of the enzymes, they're full. So the result is a limiting factor here. Third point, demonstrate an understanding of rate calculation of enzyme activity. Rate is always changing concentration over time. We will see in the analysis of the experiments. Last one, explain the importance of enzymes as biological catalysts, synthesis of carbohydrate, proteins, lipids, breakdown into sugar, amino acids, fatty acid, and glycerol. Okay, this is the topic of digestion, very important. I would suggest you to have a little summary notes of the name of the enzymes and the conversion that they do. Carbohydrate, normally we have amylase, starch into glucose. Proteins, we have proteins into amino acids. Lipids, lipase, notice all of these A's are the enzymes. It changes the proteins into amino acids. Okay, if you want to make notes, please remember to watch the video and pause it to make some notes, okay? Because I want you to hear all of from me first once, and then you can just go back to this later. Okay. Now, reflections, please do them later to understand a bit better the practical. We're going to look at two different types when we want to change the temperature and when we want to change the pH. Okay, now I will zoom out. Actually, I will look at one step at the time. So we're looking at a starch solution. So think about which enzyme are we using? Amylase. And we need to hit the temperature that is being investigated. Okay, maybe when you're looking at experiment, you already think about, oh, wait, this could be improved. Always do this analysis of improvement, safety considerations, okay? So hit the temperature being investigated, for example, like 30 degrees. Remember, this could be the thing that we're changing, okay? Second, spotting tile. Iodine is added to spotting tile, okay? This is like the instrument that it's used. It could be a bit unfamiliar, okay? But because it will make it very simple to analyze. Then we add amylase to the starch solution. Start the timer straight away. Every minute, remove a drop of starch solution and add it to the spotting tile. Observe the color. Iodine, if it turns blue-black, it's present. If there is less color change, some starch is present, some less. Repeat for different temperature. Now, I want you to take a moment to analyze these results and try to understand what does this mean. Pause the video if you need a moment. So, think about what this means. Amylase is the enzyme that breaks down starch. Of course, the quicker it will break it down, the faster, it's working, right? Now, notice we have 9 minutes, 3 minutes, and there is no color change. Okay, think about what this means. At optimum temperature, the iodine would stop turning blue-black at faster solution because the enzyme works better, right? It has broken down faster. At colder temperature or higher, at hotter temperature, for example, 60 degrees, there is no digestion because there is starch present all over. This means that amylase enzyme has denatured, so can no longer bind to the starch and break it down. Why? The active site has changed because of this denaturation. Okay? Notice at 40 degrees, it's only 3 minutes until it's all done. So it worked even faster. At colder temperature, it's not as fast. Why? As I said before, it's slowly due to the low kinetic energy, fewer collisions. Okay? This is the relevance of the experiment. And I hope you understand how easy it is to observe it with these, tiles. Okay? So, of course, there are several ways in which we can improve the experiment, but we will get some exam questions then. Okay? If you will need to look at the Corms investigation, feel free to do that with this document. The second type of experiment, and because it's the other factors, is the pH. Okay? Now, let's see the difference. Iodine added again to the spotting tile. We have a buffer solution. It's extremely important to remember this. To control the pH, you need to use the buffer solution, okay? Because this is a solution that maintains the specific pH. We had the buffer solution to amylase. Then we add the starch solution and start the stopwatch. Once again, every 10 seconds or every minute, you take a drop and put it in the tile. Every 10 seconds or 1 minute, just as long as you mention the time, until there is no more change in color. You would continue at different pHs. Like this, once again, if whenever it remains blue-black, it means that starch is present, meaning that there is no reaction or that it's still present. So take a moment. Which one do you think is the optimum pH here? It would be 5 because notice that it digests or breaks down starch faster. At pH 14 has become denatured because it is beyond the optimum. So those are the experiments that you might have done, okay? And I think they're just like quite useful to see. But remember then, the questions that you're asked could be completely different. So we're gonna get started into some examples. So we're gonna do a couple of questions, maybe similar to this experiment or different, okay? But you will see how then, you know, from this experience, you need to then be able to apply everything, okay? So this question is definitely not straightforward because, of course, I wouldn't make it easy for you today. Okay, so the first part now, you have some background information, right? Do you notice here? It's because it's a concept that you're probably not very familiar with, so you're going to have to use this information for something. Now, the marks are fatty acids are formed when lipids are broken down by lipase. And guess what? Fatty acids are acidic. Therefore, the pH decreases. And it's tricky, but I hope you understand. So these are the 2 marks. Let me put a tick. Those are the 2 marks for this first question. Okay. Second part. Describe the effect of temperature. Now, I would always, when you have to describe, you can always use the general, like S blah, 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 D blah, 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 you know, the variables. So for example, as the temperature increases, the rate of lipase increases. And then you should include some data in it, okay? So remember that as the temperature increases, the rate of lipase activity increases, here specific, and then the rate is optimal at, so it's important to mention that optimal, okay? And then you could also say that above 37, the rate decreases. Notice just a nice description using the data, okay? Because notice that it does mention the figure in the diagram, so it's important that you state that as well. Last part, explain why the activity changes. Explain why the activity changes. Okay, here is extremely important. If you just if you learn to explain it this way, you will get the mark guaranteed. An increase in temperature causes the shape of the active site, and here actually let me highlight it. 1 second. So changes the shape of the active site of the enzyme. Enzyme becomes denatured. As I said, active site, denature. Okay, and well, you can also ask yourself, so what, to make it more complete. So it does not longer function. Okay, now there is another part here, which is about the rate, and I'll just walk you through it this time, because now the rate, as I said, it's calculated as the change over time. Okay, so if you notice, there is some value missing. So from the 10% starch, we need to get like the value. No, actually, we have it over here. Okay. Calculate the rate of, actually, no, let's give it a go together. So let's have a look at the rate question, at the rate answer. Now, the rate is calculated as, you know, this change in concentration over time. Or I want you to notice something. The unit, what does that mean in terms of value? Like in terms of, sorry, equation. This will tell us that it's 1 over time. Remember, S minus 1 means 1 divided by. So if we want to calculate the rate, that's what we will have to do because we don't have a change in concentration. If it was, as I said, change in concentration over time, the unit won't be S-1, but will be unit of concentration over time. That's how we distinguish it. So you would find the mean of this, okay, which is 5 tests, so would be 117.6 as we have it here, finding the mean, and then the rate is 1 divided by, and this is the value we should get. So with the rate questions, unless it's given from the graph, like changing something over time or something, it would be check the units. Very important, check the units. Okay. I hope you got that. And now we select another question or practical. Third practical, photosynthesis. The content. Well, let's go through it again very briefly. Photosynthetic organisms and the main producers of food, therefore biomass. Remember, plants, primary producers because they make the food from, you know what, light and carbon dioxide and water. Describe the plants and endothermic reaction. Endothermic, very important to remember from chemistry, they take in energy. So they use light energy, carbon dioxide and water to produce glucose and hydrogen. Remember to write down how to write down the full equations. Now, the factors that affect the photosynthesis, the effect of temperature, light intensity, carbon dioxide, limiting factors, okay? So here, very important that you should remember like these factors and how to describe them and the reason, okay? So I would suggest you to check your notes to look at the type of diagrams that they use for the factors, because you will always see the shape, and I think it makes it a little bit easier to remember the shape, okay? An example I want to give you, it's this one. For example, notice how, again, as the rate of photosynthesis with the temperature increase. Is there any similar to something we've just seen? The enzymes, yeah, because photosynthesis is an enzyme-controlled reaction, meaning that it's affected by the same things. Okay, very important. Light intensity. This is the change. As at lower light intensity, it increases until there is something else that becomes the limiting factors. Graph levels off, because it doesn't matter if you keep increasing the light, it could be carbon dioxide or water that are the limiting factors. Same for carbon dioxide. There is always one point in which other factors become limiting, so it doesn't matter if you keep increasing the variable. Okay? Remember that that's the other things. And I think it's okay. The interaction of temperature, limiting rate of photosynthesis, we will see that in this practicals. And then directly proportional, inversely proportional, I think it's okay. We will see the diagrams, okay? Now, reflections, please go through them after you've revised this practical. And I want to show you like one type of practical, but then the important thing that I want you to notice here is, okay, here we have like a lamp and we have a ruler. So we're changing the distance of this lamp. So guess what? We're changing the light intensity. Okay. So in this case, notice that to measure, because at the end, it's about measuring the amount of gas that it's released. How do you do that? Either you have a gas syringe, but still it makes it really difficult to control because it takes in a gas, it removes another gas. Tricky. So it's useful to use. Let me highlight what I'm talking about. For example, an aquatic plant, because it, you know, we will be able to observe the oxygen bubbles, yeah, as photosynthesis occur. Now, notice that the source of carbon dioxide for aquatic plant is sodium hydrogen carbonate, okay? Notice that, because of course it's in water, but it still needs to get the carbon dioxide. Okay, and then we have this setup just because like this, we see the bubbles and well, we need to control the temperature. Okay, so this is one type of practical. Another type of plant investigation, it's when you need to investigate transpiration, but this is a different one, okay? Like with the potometer that you look at the bubbles of water moving is a different one. This is just photosynthesis, meaning that we need to have clear the process of photosynthesis to understand what we're investigating, okay? So I know this was very quick, but I'd rather you practice some exam questions for this, yeah, to apply, this type of a bit unusual experiment. And we're gonna try, I think, these first questions so that once again, you will have a couple of questions for you to revise on your own, because it's extremely important that you apply, keep practice. So this is the exam question that we do. It's just a 5-marker altogether, but notice that it is always going to be a bit different if you have like the experiment, unless you have to design the experiment that you done at school. No, you're going to have some interpretation to do. So let's take 5 minutes for you to try this. Sharing 1 second. You. Now I'll walk you through the solution of this question, because there is such an important concept that I want to highlight that you should always think about when you revise practical skills, which is, what is this? This is the control. Now, the control is what you compare your observation to see if there is a difference or not. In fact, if we want to, let me just open up the mark scheme so that you see what is the actual answer. The control variable, it's extremely important, remember, because it allows you to observe if there is an actual change or not. In fact, here, and then we can be more specific about this question, is to check that the indicator color does not change on its own, or to check any changes in color due to the organism. So very important, control variables. You always need to explain that is to check your observation, and then you would state which observation that is. In this case, would be, notice the indicator. Now, the other one is tube E, because the explanation, the most carbon dioxide, yeah, due to only respiration occurring. Now, do you understand why only respiration occurs? So it's a tricky question, because it's not only like investigating this like photosynthesis, but we're actually investigating the whole respiration process, which is possible with plants too. And I hope you understand why is that, okay? So also notice that we have a water snail there. What does that do? It does. Yeah, it does release carbon dioxide because of respiration. And then this was a really tricky question. And I've shown you an experiment before that it has some common things here, which is like the investigation of like the light dependency. But then you can be tested in like such a broad type of, like any random type of experiment like this. Okay, so we're done. So question A, just the 2 parts. Once again, because notice the weird setup of the experiment, and you just need to interpret that. So let's go. Okay, let's see if you found the similarity of the experiment before, and actually that was the fact that we have a control variable. Yeah, notice, let's see here the answers. Okay, well, first of all, the first one, I know this first one's really tricky. Again, it's a suggest question. You know that you're gonna have to think outside the line, okay? In fact, notice the solution. It's stop release of carbon dioxide, or something to mention about respiration of bacteria, fungi, microorganisms, and whatever it's in the soil. Because at the end, you're stopping the soil. I know, tricky. What is the purpose of container B? Did you notice this was another control? Once again, what is the control? To make comparison. Okay? So I would invite you to try all of the other questions of these three practicals that you have in here. Then you have additional practice for paper 1, which is like a super long paper. So just do the questions that you feel like. Practice paper 1, actual paper that you can test. Some advanced questions just specifically for Edexcel because they're a bit different topics. Okay, this equates for everyone. Okay, so you can all try. Then make sure before before the Friday paper, make sure you look at the topic list so that you make sure you've revised everything. The topic list of the practicals so that you can apply the revision that we did today to the practicals as well. Some key points, some key takeaway points of today. Analyze the type of questions that you are tested, because remember, it's there are some questions that will throw you off completely, but don't get overwhelmed by an example that you're not familiar, because it's the objective that is tested about a topic that you know, that you have to apply the knowledge to a weird example. Okay, so we've seen that. We've seen how to revise practicals. I would always suggest that you make it nice and visual, like we did today. We did just a few examples of a nice diagram. So use that, use videos. But then that's not enough, because the questions that you will be asked, like we did today, they could be relative to an experiment that is very different and very unfamiliar. But there are some common things to think about. The improvements we've seen that are always relative to the instruments, the technique, the sample, control variables. Okay, we haven't talked about safety procedures, but always think, like we do it also in chemistry so often. So I think that's relatively simple, but please try to revise all of the other experiments in this way we did today for paper 1. And we will do the same for chemistry and for the other sciences and for paper 2 as well. Thank you everyone for joining. If you have any questions, please let me know.